Hayden, I sent back your Excel spreadsheet this morning.
Yeah, I saw it. I'll just make the correction, upload it after class.
Yeah. You know, there are so many different versions of BMWP scoring. So I think it's easier to use the one that I've given you. In fact, it also has been revised recently. So there's a new scheme that takes into account actual abundances. I might put that up on Brightspace so that you have it for future reference. But what you're dealing with is the basic original BMWP scoring. Okay.
Thank you.
Green. Yeah, so do upload it to Brightspace. So I have a folder then with all 4 spreadsheets. Okay, so can you see the slide in front of you? But first of all, I just want to look at the dates for the different assignments and whether or not those are still realistic for you. So I uploaded, I hope you got the outline, the format for the report, short report. So you just work within that. So you're going to submit that as a draft first of all. Now, is March 5th still realistic? Because I know some of you on March 5th are doing the instrument presentations for the other module. So it's up to you to tell me if that's realistic. I mean, I can shift it a little bit later.
It's sorry, Mary, can I just say something? Just also that week, I also have another I have another presentation on the 3rd of March as well.
Yeah, that's what I thought. It's just there's a lot happening. So I mean, if you wish, because it makes no difference to me, I can switch it into the 12th. Give you an extra week there to get the draft done.
That would be great.
Yeah, that would be really helpful.
Yeah, thanks. So just have to remind me that I'll shift it here. So I will set up the folder then in Brightspace. I just want to note that for March 12th, and that's the draft report. Okay, and that gives me time now because I'm not going to ask you to finalize it until the 12th of April, so that those that are doing the other module will have reviewed the data, chemical data for the sites, and then it means that you can bring it into your discussion. So in the draft produced for March 12th, you talk about the quality of the sites, but you won't necessarily be able to conclude fully on what's causing problems where there are problems. So that bit can be left to be completed. So I think if you concentrate on, say, your introduction, your study area, your methods, results, and then just a few points in the discussion, and then I'll give you feedback on that. So the other and final exercise is this design of the monitoring program. So that's very important if you end up working in consultancy and you're asked to either design or review monitoring programs. So we just want to give you a little bit of guidance on that and then ask you to do an exercise. So again, we can look at this date, March 20th, and whether or not we need to shift that when I go through what is required. Okay, so you can see that is the exercise that you're going to do. You're going to design a monitoring program to detect the impact of motorway runoff into a salmonid nursery stream. There is a presentation on Brightspace that I will make visible. And that's the on-road or motorway runoff. So what you'll do is you go through that first of all, because it covers the whole range of pollutants that can come from road surfaces, because you need that information. So in terms of the monitoring program, I'm talking about maybe 6 pages maximum. So it's a short document, say 5 to 6 pages that I can scan through to get the main points on it. And these are the topics that you need to cover. So I'm just going to put that into slideshow mode so it might be a little bit easier to see. And then I'll go through it on the current slide. So that is a bit better. I just want to move this image in my way. Okay. So that 5-page document then starts with a short introduction outlining what are the likely pollutants and pressures coming from motorway surfaces, and that has to be referenced. Okay, and then based on that, you move into section 2, which is highlighting the physical and chemical determinants that need to be measured and justified. So rather than picking everything, you could come up with an enormous list. But if you're designing a monitoring program, you have to be specific to the problem that's being investigated. Otherwise, the costs are just too unrealistic. So I thought might be useful thing is to produce this table like this, where you take, say, pH, pH, and say, well, what is the source of that in relation to roads, or why would you even consider it? So what I'd say is not usually altered by motorway runoff, but can influence the solubility of heavy metals, or as you know, the form of ammonia for those that are doing the other module. And then if you come, and we'll see some of these pollutants in a moment, but for roads, heavy metals can come from just wear and tear of vehicles. So you could have a suite of heavy metals that you mentioned there that need to be analysed. Why would you do it? They're derived, as I said, from the wear and tear of the vehicles, and they're potentially toxic. And one thing you have to consider is where are you going to measure them? So it might be water or sediment or aquatic biota. Come to bioaccumulation in a moment. So just to put a noted here what you're going to do. So for heavy metals, depending on the environment, we generally look in both water and sediment, and for some biota, if we think there might be bioaccumulation of the toxic heavy metals, then we look at biological tissues as well. Then there's the biological sampling. So what are you proposing to sample? So generally what I would recommend is that you use more than one biological group. It always will depend on the budget for the project. So clearly the invertebrates are good, but fish, because you're dealing with a nursery stream here, microphytes, Phytobanthus, all of those, but I'll be coming back to them in a moment. Could be, and you could suggest them. I'm not going to put a budget limit on the project, because I'm going to ask you to try and come up with roughly some costings so that you get an indication of just the reality and the expense of some of these projects. Then the study sites will be, where are your study sites located? Now, I'm not giving you any particular motorway except to say that it's in the Midlands in Ireland, so it'll be draining in limestone. But what I mean by the study sites is where are they related or positioned relative to where the pollutants may be coming in. And in this case, the pollutants are coming in at a single point. So it's all of the drainage from the motorway is brought to a single point, and then it's directed to the surface water. So it's a point source that you're dealing with. And that influences the location of sampling sites, because they can be upstream, downstream of the point source, and they can be at different distances for the downstream. Time frame, in other words, when are samples going to be collected? So here you put in a table indicating when the invertebrates are collected and so on. But I'm going to cover those in a moment. Then the metrics. So how are you going to measure whether there's impact? So things like your Q value, BMWP, EPT, but then what metrics do we use for fish? What metrics do we use for macrophytes or phytobanthus? And then the costings. Now, come back to me when you have worked out what needs to be measured and in what, and I will give you indicative costs of each of those. Okay, so maybe to start you off thinking about that. So you will have labor costs, you'll have transport and subsistence to the locations to collect your samples. You will have analysis costs, so we'll assume that all of the analysis will be sent out to a laboratory to do. Your labor costs will include collection of the samples, delivery, or however you're going to transport them to the lab, and then data analysis and reporting. And so you'll have to think about how many days each of those might take, or at least you have to pose the questions to me, and then I compile that for you. Rather than me giving them to you, I want you to think about, I suppose, what you need to consider in drawing up the costs, and then I provide you with costs of. So say, for example, you say, well, I want to measure heavy metals in, and a suite of heavy metals in a water sample. What would that cost? Well, I'll be saying it would probably cost in the region of €40 per sample. So you need to draw up a spec of what you want to analyze, and then I provide you with the costs, and then you add up your costs. So you generate a spreadsheet. So somebody in your teams then that's good at that end of it will concentrate. Now I can keep you in the same teams as you are for the monitoring report, or for the bioassessment report. Do you want to stay in those teams, or do you want to mix around a little bit? It's up to you. Has anybody got an opinion?
I don't mind
I'm
staying
fine
in the
with my
same team.
group.
In the same group. It might be convenient because at least you're in communication for the bioassessment report. So this would follow up. Okay, where possible, tabulate and keep the text to a minimum. So the piece on the likely pollutants, we're probably talking about a maximum about a half a page, typed page. And if you do it under these headings, then we should be okay. Has anybody got any questions on that?
You'll be uploading this to Brightspace, will you?
I will. Yeah.
Perfect.
Yeah.
Thanks.
Absolutely. Yeah. So I haven't uploaded it because if you have any particular questions, remember what we did a moment, we altered the date there. So I can't actually work on sharing it. So if there's anything else I need to add to it, I'll upload this. Okay, so what I'm going to do through then is just some thoughts on biomonitoring program and some of the things you need to think about. And then if you have any questions as we go through it, just stop me. So these would be the key steps, and they're not unlike the steps that I gave you in that previous slide. So you have to define the objective. I've done that for you. Compile the information on the likely pollutants impacts. In other words, it's the profile of the discharge. And if you were working on a real case, you would also gather and integrate existing baseline information. So all I'm telling you is that it's in the Midlands of Ireland, it's on limestone. That's relevant. We'll see in a moment why it's relevant. And that it is a point discharge from a motorway. Then you need to think about any special environmental considerations. Because this is a stream or a river draining limestone, it's likely to have crayfish. So think about that. It's not in any other special area of conservation. So then define the study area, so the spatial boundaries. Are you working on an entire catchment or a segment of river? Because you're dealing with a point discharge, you're going to be working on a stretch of river. Then site selection, where you can position your monitoring points, methods, indicators, and so on. Okay, so this takes a different exercise here, but it's not unlike what you will be considering in terms of the road runoff, except that road runoff has a a wider range of potential pollutants. So suppose you were asked to detect and evaluate the impact of mining discharge on river health. Okay, so just thinking about the range of pollutants, you'd be obviously talking about heavy metals. You'd be thinking about what is mined. So generally in Ireland, what is mined is lead, zinc, but it comes with other heavy metals. They might be in low concentrations and therefore not mined, but they can potentially enter watercourses and have a toxic effect. So in fact, where you have mining activity, you have a whole suite or cocktail of heavy metals. So you have copper, cadmium, chromium, mercury, and so on. Particulate matter, organic matter, if you have, say, sewage treatment works in the area, and in the case of mining, you can often have nitrate generated from the use of explosives. You have to think about the full range of contaminants and where they're coming from, so that you don't miss any of them. So you think about the whole process. So for roads, you'd be thinking about, what accumulates in wet conditions, what accumulates in dry conditions. Then what are the components of the system likely to be impacted? So you have to think about the physical environment, the chemical environment, and the biological environment. So the physical environment, it will be impacts on hydromorphology. So the major one would be siltation or contamination of sediments by heavy metals. Chemical then, so these are the metals or whatever contaminants are in solution. So you'd have the contaminants, in this case toxic metals, but also then might they affect sort of the general condition of the water? So do they affect changes in pH, oxygen, nutrient status? Then in terms of biology, as I said, it's good to include a number of different, what we call the biological elements or biological groups. So invertebrates are always there because we have good metrics. But there's also the vegetation and fish. And then in terms of mining, we often look at contamination of fish tissue. So bioaccumulation of metals in tissues. Now, study sites. So you have a study area. So your study area is the Midlands of Ireland, somewhere. Now you need sites on your rivers. Because it's a point source, you can have controls upstream. And generally, we recommend that you have at least two control sites upstream. If you think there's input from some other source upstream, then often we consider having a reference site on a separate comparable river. In the case of your study, road related, there's no other issues upstream. So we limit you to control sites upstream of the point of discharge. And then generally we have sampling points downstream of the point of discharge. And the first is always immediately downstream in what we call just outside of the mixing zone. So you make sure that the effluent can be fully mixed before you sample, so you get the effect of those contaminants. And then at varying distances downstream, if you think the contaminant is likely to be transferred further downstream. Now for you on this particular exercise, it's a sort of an exploration, so that you know particular background information, you don't know how far the contaminants are transported. So it might be useful then to have a number of sites, say 100 meters, 200 meters, 500, and going, I would say, not much more than 1 kilometer or even 1 or 2 kilometers downstream, because the difficulty of moving downstream is that you can encounter other sources of pollution that can complicate So you might just put a note into that effect, you know, that the other, I suppose the last downstream site will be determined by the possibility of other sources of pollution. Okay, so that's your sites. Any questions on those? Okay. So then it's your indicators and your metrics, you know. Any of these things will indicate something, but they're not always going to indicate everything that you have an interest in. So you have to think about what you measure. It must be relevant. You must know that they respond to the stressors and must be easily measured, and they must integrate over time. So that's what your invertebrates do, and that's what makes them very relevant to water quality determination. So as I said, it's useful to select multiple indicator groups. So in your biology, it could be your macrophytes, your microinvertebrates, fish, macrophytes, and also Phytobanthus. I'm going to mention Phytobanthus later on. So that's the biofilm that's on rocks, and it can be very good indicator of sometimes acidification, but principally eutrophication, as can microphytes. So you might decide which of those vegetation groups that you want to work with. Measure suspended solids, degree of siltation. We've covered sediment. Now with chemistry, we have to think about what medium. So obviously water, you have pH, alkalinity, oxygen, sort of the standard ones. Nutrients, if you think they're relevant, but you're going to have to justify nutrients as to where nutrients and contaminants might come from a road surface. What on a road surface might release those? But we're dealing here with mining, so I would say nutrients. Then heavy metals, we've mentioned those. If you're dealing with an area with that's alkaline, for example, drained limestone, then some of those metals are likely to accumulate in the sediments. So they don't necessarily remain in solution. The more acidic the water, the more potential for metals to remain in solution. But in this case, we're dealing with heavy metals. A lot of those can accumulate, again, because I'm dealing with limestone area that can accumulate in sediments. Now, when you look at contaminants from roads, you're going to come across a contaminant known as PAHs, polyaromatic hydrocarbons. And they, regardless of chemistry, have a tendency to accumulate in sediments. So just note that you will be analysing sediments for that, or at least you'll be recommending that they're analysed for the PAHs. So think when you're going through the presentations, what occurs in water, whereas in contrast to what might occur in the sediments. Now, one of the things I asked you to think about is the timeline. In other words, when would you recommend samples be collected? For invertebrates, the ideal, if finance allowed it, is to collect in 2 seasons. Spring, because that'll capture what has occurred during the winter into the spring. The value of the spring is that invertebrates, you've got the greatest diversity of invertebrates, they're at their largest size. And then at the end of the summer, so into the autumn. And that's useful because that gives an indication of what has happened over the summer months. And that's the period of greatest stress when you have the lower water levels and you have slightly higher temperatures. Now, the sampling for invertebrates, as we've already mentioned, can be single habitat or multi-habitat. The tendency nowadays is to go for multi-habitat. I'm asking you to justify your choice. The reason I'm saying multi-habitat is that it allows you to encounter the widest range of potential impacts. So you might have fauna that live in other habitats, apart from riffles that could be impacted by pollutants. Now, microphytes are sampled generally once a year. So these are the large visible plants, watercress, that sort of thing, water rosefoot. In the summer months, usually between June and mid-September. The reason being is that the plants will is large enough to examine, but they'll also have their flowering structures on that during that period. What I haven't on there, and I'll just mention it, is Phytobanthus, or what is called periphyton. So that's the biofilm on the rocks. And here again, depending on finances, there's 3 approaches, 3 times that can be sampled. Sampling in the springtime, generally around April, water temperatures increase. The autumn time, September, October, but sometimes the sampling is simply concentrated in the summer months. I prefer the spring and the autumn sampling periods. Protected species will be your crayfish. So we went through the sampling of the crayfish. You used your trappy traps, and normally what we use is 10 trappy traps placed per side and left for 24 hours. And then you have your measurement of the length and the weight and the numbers in the traps, expressing them as catch per unit effort. So catch per trap per day. Physical evaluation of the habitat conditions, so the usual thing, looking at your representation of your riffles, your substrates, but also measuring siltation. Now the chemical, the recommendation for most is monthly sampling. In the case of mining, they usually have continuous measurement of some determinants, but I think for your exercise, monthly sampling is enough, except you might have some sampling targeted at times flood events, and flood events known as the first flush effect. And the first flush effect is first rain after a dry period. And the reason for that is that during the dry period, you get a lot of pollutants accumulated on the road surface, and then when you get the rainfall, those can be transferred into surface water. So you're getting the worst case conditions. So you could target monthly sampling and then some samplings, some what we call event sampling. So targeting the first flush events. Okay, any questions on that? Okay. There was a report. Yeah, somebody has a question.
The Phytobanthus, how many times should we, I know you mentioned 3
I think
times.
spring and autumn.
Spring and autumn. Yeah. Thank you.
Yeah, I'm giving you references later on to some manuals. So maybe looking at the fish. So obviously lots of different fish species, but most of the effort nowadays is concentrated on the salmonids, but the occurrence of other species is also noted. The exercise I'm giving you is on a salmonid nursery stream, so I think you're going to concentrate in a collection of metrics for salmonids. Now, just to remind you about the WFD, and that required us to look at species composition. So in other words, you record all the species present, but you concentrate on assessing the abundance of what are the sensitive species, which are the salmonids. So here we're talking about the salmonid stream. In this case, is mainly brown trout. Age structure and reproduction, that will be in terms of, you measure that in terms of recruitment of the juveniles. So when you look at the age structure of your population, you'll see whether you have juveniles. So that will be the measure of that aspect there. And each of those can be described by different metrics. So I mentioned the metrics. Now, there's a difference between sampling for fish say invertebrates. So fish sampling requires a larger spatial scale. So with invertebrates, you define your study area as generally 30 to 50 meters in length. The fish sampling length is defined as partly by the width of the stream. So the general guidance is to pick a length of stream that is 10 times the wetted width. And then the method supplied must be specific to rivers, because it's quite different between rivers and lakes. And the reason we include different indicator groups is that they're all not necessarily equally sensitive to the same stressors. So it allows you to detect impact of the widest range of stressors. You have to bear in mind that there are factors that naturally affect fish numbers, such as habitat, food availability, natural predators, and droughts and floods. So having a drought period at the beginning of the year can reduce the amount of wetted habitat available for juveniles, and we've seen that in the Y water. Or floods during the early spring period can wash out the eggs from the fish legs. And then you add, so what it means is that naturally you have a small percentage of juvenile fish surviving to the end of their first year. That's generally less than 2%. So your typical average brown trout would have about 500 eggs, and only 2% of those will become 1-year-old fish. And even less than make it to the end of the first year when you have all of these additional pressures. And that's just a selection of those that you'll be fairly familiar with. Okay. Now, there are also different factors that affect the distribution, composition, and abundance of fish, and you just at least to be aware of those. So altitude. So obviously trout will go to a certain altitude, but their numbers diminish with altitude. The wetted widths then determine how much habitat is available. The depth will determine what size of fish occur, and the same with the flow, the gradient, distance from the source, and so on. So you need to have these descriptors, to be aware of those and have them in your site descriptions. Okay, so fish then in rivers are generally sampled by electrofishing, passing a current between the anode and cathode. Where the river width is wide and relatively deep, the electrofishing gear is what we call boat mounted. So we have a generator generating a current which is passed between, you see, one will be your cathode and anode there, and fish in the general area of that can be stunned and caught. If it is a shallow river, that is done using backpack electrofishing. So you're carrying the unit on your back, and you have in your hand then the cathode and anode. Okay, so now you have your fish. So you have to carry out a number of measurements. So you will refer to these in the report. So generally what we measure is the fish length from the tip of the snout to the fork of the tail. This is a brown trout here. That's a wild brown trout. This is one of hatchery origin. And I recognize that because you have damage to the fins. Fish length is also measured. And we also take a sample of the scales of the fish by simply scraping the surface there. And that's usually taken from what we call the shoulder, this region here. So you'll be saying that fish will be measured, weighed, and samples of scales taken for age determination. Okay, that's just some of the bigger fish that we get, sea trout. So how do we measure the age of the fish, well, similar to aging a tree, in that you have these annual rings. In fact, you get rings that are continuously laid down, but the spacing of the rings varies depending on the growth of the fish. So what you end up with are these checks on the growth. So during the winter period stops feeding if the temperature drops below 10 degrees, and therefore the distance between these growth rings lessens. So if you look here, widely spaced, so this is a fish that was hatched, say around March, then coming into spring, summer, you get good growth, and then growth eases off in the autumn into the winter. Into the next year, you see the good growth, and then you see a check on the growth, and then you're into the next year, good growth during the summer, spring, summer, and then a check on the growth. So you can say this is the end of its first year, the end of its second year, third year, and then there's a little bit extra out there. So that is what we call a 3+ fish. Okay, so you're looking for those tightly spaced bands. You can see the checks along the edge of the scale there. This one there, 3+. And what we can do then is take a measurement of that distance to each of the annual rings, what we call them, and we can use that measurement to back calculate the length the fish was at the end of each of the years, and then use that to show the growth profile for the year. So in your report, you'll say that the fish will be aged using the scales, and back calculations will be performed to estimate the length of the fish at the end of each successive year. Okay, there's another one there. You can see the rings. Now, so generally we use these scales, but fish living in very acidic environments can have regeneration of the scales. So you see areas that are quite damaged on the scales, and it's difficult to age the fish. So in those circumstances, and it's very seldom, we use the otoliths, which are the ear stones, but it means sacrificing the fish. But that's seldom done. The same applies here, is that you get these bands laid down on the otoliths, like we use to age the fish. Now, I mentioned that you measure the fish, you can age the fish, and then it allows you to illustrate the length frequency distribution. And if you have aged the fish, then you can apply an age to each of the cohorts that you see on the diagram there. So these are your juveniles, your 0+, your what we call 1+, and 2+, and then the older fish. And for salmon, or for a salmonid nursery stream, you would most likely see a profile like that. So it should have the highest abundance of the juveniles, and then the numbers diminish as they get older. Partly because some of them will move to find more suitable habitat. And in fact, you probably get quite low numbers of 2+ and possibly no 3+. So that's one way that we illustrate it. Sometimes we don't color code them as clearly as this, because while the juvenile group are easy to distinguish, as the fish get older, you can have overlap between the age groups. So you can have some 1-year-old fish that are fast growing and others that are slightly slower growing. So you get overlap at these points here. So what we normally do is we draw a line there indicating the extent of the, say, your 1+, and another line showing the 2+ and where there's overlap. So when it comes to collecting scales from fish, we concentrate our efforts where we know there is an overlap. So I would be collecting a lot of scales so just around this area here, in case we have, you see this fish here that has, it's only about 4 centimeters. That fish at the end of its first year, going into the second year, is probably not going to be more than 10 centimeters. So there might be a little bit of overlap here. So concentrate on getting a lot of scales in that region here, and especially here, because as they get older, you see the overlap here. There's some 3+ fish in that region there. You get a lot more overlap as they get older, because that growth difference in the first year gets exaggerated over time. Okay, so you will draw then length frequency distributions to illustrate the population structure. That's what I'm showing there. So you have your composition of your catch, all the species, and the population structure of your salmons. And so I'm telling you, there's only going to be brown trout in this river. Okay, so how do you measure abundance in rivers? The usual method is using the successive or depletion method, but it's also possible to do market capture. The only problem with market capture is that you have to mark a very large number of fish in your population before you can get sufficient recapture to estimate the population size. So that method involves going out sampling and marking the fish that you've caught. That can be done by fin clipping or dye marking. Then they're released back into the stretch that you took them from, and you come back on another day and resample, and based on the proportion of fish in your recaptured sample, you can estimate the population size. And then there's gear calibration, which sort of gives an estimate of the percentage of the population that's captured using a particular method. But I said the usual method is to use this depletion approach. So it's known as the successive removal method, and it's based on the principle that if you fish a river in number, or a river stretch a number of times successively, the number of fish in each sample decreases, and the rate of decrease then allows you to estimate the population size. Now, you can, the number of times you fish the section, so this is all done on one day. So you fish the section, you hold the sample of fish separately, you fish it again, you have a second sample of fish, you fish it a third time if it's needed, or a fourth time if it's needed. So if it's needed, depends on whether the numbers are depleting. If you can catch, and you can do rough calculations as you're doing this, if you can catch, say, 80% of your estimated population in the first catch, then one other catch is only needed. So if I caught 60 fish in my first sample, and I caught 10 or 20 in my second sample, I would say there's no need to do herd fishing. And if I caught 60 in my first, and I caught maybe 35 in the first, I'd say let's fish it again just to get the depletion. So you're getting a good line of depletion. So you have what is called a two sample approach, or a 3 sample approach, or multiple. Now, each of this method is subject to a number of assumptions. So number one, there shouldn't be any immigration or emigration during the sampling period. In other words, the fish, you don't have fish coming in and out of your sampling stretch during that assessment period. And the way we achieve that is that we position stop nets at the end of the sampling reach, so the fish can't move in and out while you're doing the sampling. Individually, the fish must have equal probabilities of being caught. That's generally it's fine with electrofishing. And all members of the population must be vulnerable to the gear. And then a significant portion of the population must be removed during each fishing. So be it a 2-catch method, or a 3-catch, or a 4-catch. So for your stream, you will be saying that we will use the successive removal method based on either 2 catches or 3 catches, depending on the number of fish caught in each, and that the study reach will be isolated with stop nets during the assessment. That's the sort of wording that you put in there. Okay, that's how to quantify it. Now, in some rivers, and that's the ideal there. You can see that the stop net is fixed nice and firmly to the bottom of the river. It's held up. But in some rivers, it's quite difficult with a fast flowing to set stop nets. And occasionally in those situations, we simply fish from riffle to riffle. And the riffle acts as a sort of a barrier. Now in lakes, you can have electrofishing in shallow margins, but normally what is carried out is gill netting and using macro capture methods. Gill netting allows you to express the numbers per unit area, or per catch per unit area, or unit effort, depending on what we're dealing with. Now, heavy metals. As I said, they have to be analysed both in water and sediment, particularly in calcareous areas. The heavy metals will end up in sediment. Must think about animal tissues for bioaccumulation. Now, we have worked with 2 animals for bioaccumulation studies. The first is the gambits, so that's your freshwater shrimp. Very useful because these live quite close to the sediment and consume a lot of organic matter in the sediment, so they have potential there to uptake heavy metals. So with those, you collect, so the standard method is to collect your gambit live, hold them in the laboratory in an aquarium for at least 24 hours to clear the gut. The reason we're clearing the gut is that it clears it of any sediment, material that's not being digested, and that could give you a wrong result, contaminate your sample. So that step must be undertaken. And then they're sent off for analysis. So the whole animals are used for analysis. So my sample size is not an individual animal, but it is generally about 50 gametes. So with your sites, if you're going to look for a bioaccumulation, if you think heavy metals are an issue, you collect 50 samples of gametes at your study site, and then they're treated as a single composite sample. And they're sent off then. So you collect them, you allow them to clear the gut, they're sacrificed, and then sent off for analysis. Now, with fish, with the trout, normally we would be looking at heavy metal contamination of trout, where those trout are likely to be caught for food. But if you also wanted to see the condition of the fish, if you thought their condition might be impacted, or the health of, say, your juveniles might be impacted, then you could also look at heavy metals in the smaller fish as well. When it comes to fish, you have a choice of different tissues that you can work with. If it's fish that one is interested in terms of angling, we always look at the lateral muscle, because that's the bit of the fish that you eat. The gills and eyes tell you something about the external environment, so you can have accumulation of heavy metals in those, particularly in the gill. Gills are very useful because that's where you have the filtering of the water. But internally then, the liver and the kidneys tell you about accumulation in the body as well as the lateral muscle. So if you wish, you can include some analysis of trout tissues. Only thing to be aware of is that you pick fish within the same size range, or you have samples within 2 different size ranges, because the amount of metal accumulation increases with fish size. So if you took small fish upstream and took big fish downstream, you wouldn't be able to say that it was due to the pollutant coming in. It might just be natural accumulation as the fish gets bigger. Now, okay, so you've done your sampling, you've done the analysis, you've sent it all off. So, and you now have data back. So you have to calculate metrics that are going to tell you whether there is impact. So always the best thing here is to use multiple metrics where possible, particularly for invertebrates, because the different metrics will tell you different things. And you're going to have some heavy metals coming in from the road's surface. So it might be worth doing a little bit of a search and see if there's any metrics, invertebrate metrics that are specific for heavy metals. Q value should pick up some of it. So you have your number of taxa, your taxon richness, taxon abundance, because of whole community abundance can be affected. So you need to measure those. Your EPTs are important, your biotic indices. So that's your invertebrate suite of metrics. Then fish, you're going to look at your age structure of your population, the density and the condition. So recommend that you look up what is known as condition factor. A way of measure, and then to put it in there for you. Condition factor should go in there. I'll just skip out of that one minute. It's a metric, and it's a metric that expresses the relationship between the length of the fish and the weight of the fish, and is therefore a measure of how fat or plump the fish is. And that's useful to measure. If you decide to include microphytes, then there is an index known as the mean trophic rank index. And that's a link. I can't actually get the manual, but that's a link to the manual. And in particular, page 33 will give you the formula that you use, and 36 will show you an illustrated calculation of the net of the metric. It's not unlike invertebrates in that you have list of species of varying sensitivity, and then you take a measure of their abundance in the environment. So that manual also shows you the method. So with microphytes, what you're doing is measuring the percentage cover of the different species. So you move up the screen in a zigzag pattern and measure, or you record the percentage of the different species present, and that's carried out during the summer months. Then there's Phytobanthus, or what we call also periphyton, and so that is the biofilm. So it's a matter of scraping some of that off cobbles, and the standard approach is to use a toothbrush to scrape the material off 5 cobbles. And the index then is based on the diatoms contained in that sample. And I've given you a link here to the index. The index calculate is mentioned on page 31, the formula. Page 39 has a worked example. What we normally report is the trophic diatom index, but you can also convert it into a diatom quality index, which is essentially 100 minus your TDI. So the TDI is the one that I think you will be calculating. And I think I want to remember, if your index value is less than 50, then you have good conditions. If it's greater than 50, you're dealing with impacted conditions. Both microphytes and Phytobanthus, those indices are useful for detecting eutrophication. But I think the trophic diatom index can also be useful where you might have other contaminants present. So, I mean, the other measurements then, the bioaccumulation will be your concentrations of your different metals relative to your control. Okay. Can I put this up? So that's an example of your mean trophic rank index. So you have sensitivity of the species, you have a cover value, And it just shows you then how you calculate the index. And generally with the MTR, your value, I think it's different from the trophic diatom index. If you get values greater than, say, 65, it's unlikely to be eutrophic. But if you go to those pages that I recommend, you get enough in that. So overall, before we finish, and these are recommendations that come from the US EPA in terms of monitoring programs. So you deal with multiple assemblages, so more than one group. That's what I'm recommending here. You don't have to include every group. You can pick a few. That you have your habitat assessment. You use standard operating procedures. That's where I went through the methodology. And you have some sort of quality assurance there. So in other words, if you're sorting samples, that there's some assessment of the how well they're sourced. Multiple metrics that strengthen your data interpretation, particularly where they're across different groups. Regional reference, so I'm saying your upstream site is fine, there's nothing happening upstream, so that will be our control or our reference period. Then they also recommend that you have a set index period, so that means that you're collecting the data each year. It's an annual event in the same time. That minimizes the effect of, say, season, for example. So if you say I'm collecting the invertebrates in April, I'm collecting them at the end of September. I would say in Ireland, invertebrate summer or autumn sampling should not be earlier than the end of September. The reason for that is that a lot of the invertebrates that have been are in the juvenile, very early juvenile stages. So something like Mithridina, one of the mayflies, you will not detect the new generation of those in early September. It'll be the end of September before you see those. So end of September, beginning of October is a good time in this country for collecting autumn invertebrates. And that's just the same. Any questions? Now, when you start doing this and you have any questions and you think a Zoom session would be useful, just send me an email and I can set that up and discuss the questions you might have as you begin to design your monitoring program. And it might be a useful way to cover the costs, because you then will know what you want to sample and I can give you some idea of the costs. Probably best to do it with the full group rather than individual groups coming up. Would that suit most of you?
Yeah. How many, so like say we have the macroinvertebrates, we've fish, we might be doing plants as well. How many would, or is that like, how many should you be wanting to analyze? Or would we kind of come up with a big list and then get the costing and then maybe have to pare it down a bit?
Yeah, well, you won't have to pare because I'm not going to limit you on the costings. I think if you can make a list that you think best covers the problems that might be encountered. So I don't mind how big the list is. And in fact, if it is as big as possible, then it gives you, I suppose, the experience in looking at those groups and how you might sample them and so on.
Okay, thanks.
Yeah, but I think you're going to be keeping it quite short. And always the main thing is that you justify why you want to target a particular group and why you want to target multiple groups, and especially for the chemistry, why you want to have certain things analyzed. And you discover that road runoff has a lot of potential toxins in it. So maybe the first thing is to make it visible immediately is to have a look through the presentation on road runoff. And then you can work from there.
And are we picking a stretch of motorway ourselves in the Midlands? You're assigning it.
I'm just saying you don't need to know where exactly it is. All right, you need to know that, it's in a limestone area.
Okay.
There's a point discharge from the drainage. The drainage is coming to one point, and that's normally how they're built, is that each section will have a catchment, and then it discharges sometimes through the filter bed. But even if it's through the filter bed, it does eventually end up in the stream. And we're seeing here that it's coming in at a single point. And I'm also saying that upstream, there's no pollution problems. Yeah, so you don't have to go and find a site. I'm giving you this potential location.
Okay, thanks.
All right, anybody else have any questions?
Yeah, so it goes through a filter bed, and then through a pipe.
And then it runs into the river after that, yeah.
Okay, at that point, it's not going to diffuse through the river?
No, it's not diffused. No, it's
Okay.
all brought to one point and discharged after that.
Grant, thanks.
So you can't assume that the filter bed is doing anything, so you're going to look for the contaminants. So we're assuming you know nothing about, about these potential contaminants in the water. Okay, so overall it'll give you, I suppose, a little experience in designing a management program and costing it. But obviously in the real world, you'll be narrowing down. That'll be defined by the budget. So at least you can say you have a little experience in costing. Okay, and that would be useful to do it on a spreadsheet. So maybe the whole costing, so when you submit it, the 6 pages, if you also submitted your spreadsheet that you had your costings on, but that you could provide a summary of them in the Word doc. So you might have labor, travel subsistence, and external analysis, so on. But the labor can be broken down into field work, data analysis, reporting. Now, I do know we will need another session because you will come back when you design your monitoring program. You'll probably ask me, how long would it take me to collect samples? How long would it take me to analyze the data? You have a little sense of that because you've calculated some metrics. Now, if you were to double the metrics, how long would it take? And then reporting, how many days would one allow for reporting? Okay, Hayden, did you have a question there?
Yeah, sorry, it was actually about the policy brief. I just wanted to wait till we were done about this.
Okay, are we done on that, or does anybody else have questions? So just give me a bit notice if you want to meet again to talk about timing, sort of the length of time it takes to do each of these bits, and then the costings. And so you communicate among yourselves, yeah?
At the date, are you keeping the same date?
I mean, I need you to go away and do your reading before and do your, I suppose, your outline of what you think you'd want to sample. That needs to be done before you can come and ask me questions. So it might be in 22nd next week. So it might be
for the deadline.
Okay, let me need to get that done. Let's go back to the deadline. It's 20 something. 20th of March, is that reasonable? So we probably need to meet, depends on how quick you're going to do this, because you're doing the bioassessment draft report first of all. So if you wanted to leave it until later in March when you're nearly there, because that's going to be the last exercise. And until you do it, So you could be, date I put there, 20th of March. Maybe the week beginning the 15th. I know it's the break period, but I can meet you then. I think go away and see your own timing and see how the group is timing the work, and then come back to me with a suggested date.
But you think the majority of the work we'll have done before we have this second Zoom with you?
Yes, we
Okay.
need to have that done, because the second Zoom is talking about, well, how long would it take to do each of the tasks and the costing. So you could have your spreadsheet and all set up, or I'll design a spreadsheet for you, and then you can work on that. I think that's the best and most efficient use of time. Unless you have certain other questions, but a lot of them I can deal with by email. That's just probably the hardest part of the end, is that you have least experience in, is knowing how long it takes to do anything like that, and then costing it. Okay. Any other question on that before I take the policy brief question? No? Okay, policy brief, Hayden.
I just had two questions. So, one, did you want a title page, or is it just the one page only?
Just one page.
Yeah. Okay. And then two, just to confirm that it's simply just talking about 5 reasons
Yes.
that you're persuading the government to protect, not anything about legislature or anything like that.
No. No.
Okay.
Well,
I
I
just
mean,
wanted to confirm.
if you wanted, one of the reasons could be that we have legislative requirements,
Mhm.
policy
Okay.
requirements, yeah, and then add something else to it. If you can link it to benefits for not only the environment but people, that can make it stronger.
Okay. So like room for improvement too in gaps and.
Yeah.
Okay. Thank you.
Okay. Anybody else? And again, just look at how you want it to look. So I'm not just going to say 5 bullets. You can put a bit of design into it if you have the time to do it. Make it attractive.
And would you like it formally addressed to like a TD? Like
No, because a policy brief can be broader. So it's called a policy brief, 5 reasons, and then put the reasons down.
Thank you.
Mary, can I just ask you something? It's about the Excel, about the bioassessment Excel sheets that we were doing.
Yeah.
Yeah, sorry. Did you receive the one I sent you? Like, I sent asking you a question. We weren't sure if we had fixed the problem. Did you receive that?
Can you send it again? You're not in Hayden's group 1, are you?
No, we're group 4.
Okay, send it again. I'll have a look at it.
Okay.
Yeah, because there was a problem with calculation of the BMWP for the Glencullen, and I think it was the calculation of group C on something.
Yeah, we think we fixed it. It's just that I sent it to you to ask, like, oh, was it fixed? I didn't response. I didn't know if you got it.
Right. Send it now, and I'll look at it and get it back to you.
Okay.
Right. And then what I recommend is that you will put up the spreadsheets onto that folder on Brightspace, that we have it, so we have the completed spreadsheets.
Okay.
Okay. Any other questions? You have a lot to do now. You're busy 2 weeks. Still, on terms of the other module, I'm just waiting for responses to whether we can run the labs in the break. So I'll come back to you on that. But in the meantime, I am going to compile a set of chemistry data, and you'll work your report on that. And then that knowledge will feed into your bioassessment report as well. So So hopefully that both parts will come together. All right, leave it at that then. Okay, and I'll upload this presentation. Okay.
Thank you.
Thank you. Bye.
Thank you.
Thanks.
Thank you. Have a good weekend.
Thank you.